Good afternoon students, myself Mrs. Ashwini Kadbhane from Department of Microbiology, S.V.K.T College, Devlali camp. Today we are welcome to the online lecture series. This is my 15th lecture of the LMS. So today we are going to see the SYBSc paper number 1, that is the genetics. In our previous lecture we have already seen about what are genes and why it is required for the duplication of the cell. So in our today's session we are going to understand the DNA, and we are going to learn about the history, how the development and various experiments prove that the DNA, it is the only genetic material of our body. So here we are going to start our today's lecture. Before starting our today's lecture, we are going to revise what we have already learned in our previous lecture. So let's recap. We have learned about the biomolecules, which are the essential molecules of any live cells, which includes the proteins, carbohydrates, lipids and the nucleic acids. So essential components of the cell, which we have already learned. We have seen the introduction about the genetics. We have seen the mitosis and meiosis, the difference between the mitosis and meiosis. We have seen that in the S phase, there is going to happen the DNA replication. Okay. So all this we have already seen, and the Mendel's finding. In our today's session, we are going to see the search of genetic material, how the genetic material was searched, what is the initial discoveries and history of the DNA structure. We are going to today our focus is on two experiments, that is the Griffith experiments and the Avery Macleod experiment. The Griffith experiment is also called as the transformation experiment, and we are going to see its conclusion. We are going to see the next experiment, that is the Avery Macleod and McCarty's experiment and its conclusion. Okay. So today our focus will be on these two experiments, which are the experiments which prove that the DNA is the only genetic material of a cell, which is passed from one progeny to the next progeny, or from the parents to the daughter cells. So the search of genetic material, some substances must responsible for passage of traits from parents to the offsprings, and that passage of traits, it is your DNA, which the experiment and various scientists prove that is not a protein, that is not a carbohydrate, that is not a lipid, it is only the DNA, it is not a RNA also, it is only the DNA which is passed from one progeny or one parents to its offsprings. Some viruses, they are having the genetic material as RNA. Example, TMV virus, it is having the RNA as a genetic material. So in that, the RNA is playing that role. But otherwise, in all cells, the DNA is only the genetic material which is passed from the parents to its offspring. For this, a substance which is a genetic material, it should be stable enough to store the information for the longer period of time. It have the ability, it should be able to replicate it accurately. It should capable to change and to allow the evolution. As we have seen that the evolution, because of the evolution, many new characters have been accompanied by us, by humans also. First, we were having the bended portion. Now we are standing up very straight. Because of the evolution, the genes have been evolved, and the various functions of our body, it has been accompanied by our body. So evolution is merged by the DNA. We have already seen the Mendel's finding. So his finding suggested that there was a factor which was transmitted from a parent to its offspring and inherited. He has shown us the pea plant experiment and various experiment which show that there is one factor, but he can't prove that which factor is that which is being transmitted from a parent to its offspring. So this factor was not understood till the 20th century. After 20th century, many scientists came and they explained it was the DNA. It was the Frederick Griffith, the first experiment, who told us that it is the DNA which is transmitted. So Mendel's principle, they are the laws of segregation, laws of independent assortment, laws of dominance. All the three laws, They are being explained by the Mendel for the genetics. So what constituent of the cell carries the genetic information? They are the genes which are highly stable requirement yet capable of permanent sudden change to mutate forms to provide a basis of evolution. The genes, they can be mutated, there can be mutation, it may be spontaneous or it may be an induced type of mutation. If I am exposed to a sunlight, my body adapt that, and because of the adaptation I have the sun tanning or various such problems. That is not a mutation, that is the adaptation by my body. If the genes, they are getting mutated, then there would be a different character which may be seen in our offsprings or which may be seen in the progenies. So that is the mutation, that a gene can mutate it and can have a sudden change in it. Proteins, strong evidence initially in favor of proteins. Garrod and Tatum, they were the scientists, they proved that in the theory of gene that enzyme controlling it is done by the protein, and protein, it is the carrier of a genetic information. But after that, Griffith experiment, Griffith experiments, and later Avery, Macleod, they proved that it is not a protein, it is the DNA as the genetic material in the live cells and some phages and the viruses. In RNA, it is RNA in some, as I told you, in some viruses, example tobacco mosaic virus, it is having the RNA as a genetic material. So in some cell, RNA is carrier of a genetic material. Now we are going to see as DNA as genetic material. DNA was first extracted from the nuclei in 1870 and it was named as nuclein after their source. Their chemical analysis determined that DNA was a weak acid and rich in the phosphorus, because because we know that DNA is compromises by the nucleotides, and nucleotides, they are made up of the nitrogen bases, a sugar molecule and a phosphorus molecule. So it is rich in the phosphorus. Its names provide us a lot of information about the DNA. That is the deoxyribonucleic acid, which contain a deoxyribose sugar, which is the 5-carbon sugar. It is the weakly acidic and is found in the nucleus. Because of its nuclear localization, subsequent identification, it is components of a chromosome and it is a carrier of the genetic information. So bacterial transformation implicates the DNA as the substance of a gene. So in 1928, Frederick Griffith experiment with the smooth and the rough type of colonies. Smooth means virulent type of colonies which can cause, which is a pathogenic bacteria and which can cause a infection in us. Okay. Virulent means a strain which is capable to cause a disease. Avirulent means a strain which has not a capability to cause a disease, which are harmless, and virulent which are which can cause the disease, means pathogens and non-pathogens. So he selected Streptomyces pneumonia, Streptococcus pneumonia as the organism, and in which he isolated the two types of strain, that is the smooth and rough. Smooth one which were having the virulent type of strain, which was the pathogenic strain which was able to cause a disease. And why it is smooth? Because it is having a capsule on it. Because of the capsule, it is a looking a smooth colony. And non-virulent, that is a protein coat, they are not having that protein coat, they are not having that capsule, so they are rough type of colonies and they are non-pathogenic. Bacterial transformation demonstrate transfer of the genetic material. This was done by the Frederick Griffith. And in 1944, Oswald Avery, Colin Macleod and McCarty, they determined the DNA transformation material by their experiment, and they proved that it is the DNA only which is the genetic material. So identification of the DNA as a genetic material. To fulfill its role, DNA material should have the several criteria. It should have the information, it should be able to transmission, it should be able to replication, and it should be able to variation, that is capable to have a change. So information, it must contain the information necessary to have an entire organism. It should have the entire information of that particular organism. all the genes, all the character which is present in that organism should have in that particular genes. Transmission, it should must have the capability to pass from the parent to the offspring. A replication, it should be copied, means a same duplicate copy should be, it should be able to make. In replication, various enzymes they are required. Example, your ligase, gyrase, polymerase 3, polymerase 1, helicase, all these enzymes are required. So it should have that capability to pass from parent to the offspring, and variation, it must be capable of changes to account the known phenotypic variation in each species. Now, history of DNA structure. George, now we are going to see about the history in genetics. What were the histories before the Frederick Griffith, before they do the experiment, what were the histories behind the genetics? George Mendel in 1857, he was the father of genetics because he showed the inheritance in the pea plant, but he was unknown about what it is passing from the parent to its offspring. But he was knowing that there is something which is passing from a parent towards its offspring, and they are carrying some information about the characters. Friedrich Mischer in 1884 to 1895, he discovered a substance called the nuclein in 1869, which was a unique ratio of which was having the nitrogen and the phosphorus. Later, he isolated a pure sample of material now which is known as DNA from the sperm of the salmon. In 1889, his pupil Richard Altman named its nucleic acid. This substance was found to exist only in the chromosome, so he called it as the nucleic acid. Then came the Frederick Griffith, a scientist who was working a project in 1928 that he formed that the basis that the DNA was the molecule of the inheritance, which was shown by the George Mendel. In 1929, Phoebus Levine at the Rockefeller Institute identified the components of the DNA, which were the molecules of the 4 nitrogen bases, that is the adenine, guanine, cytosine and the thymine, and the other molecules, that is the sugar, that is the ribose and the deoxyribose sugar, and the phosphorus molecule. They are present in the DNA which was identified in 1929. Now we are going to learn about the Griffith experiment, which was the first experiment to show that the DNA is the genetic experiment. So what did Griffith did? He took an isolate which was of the Streptomyces pneumonia. He took the 2 strain of that, which one was smooth strain and one was rough strain, and he injected that 2 strains in the mice. Frederick Griffith in 1928 experiment with the Streptococcus pneumonia bacteria in mice showed that something passed from the dead bacteria into the nearby living ones, allowing them to change their cell surface and cause the disease and cause the mice to die. He called this agent as the transforming principle, so his experiment was known as the transformation experiment, but did not know that what it was and how they worked. See, in the figure you can see that he is the Frederick Griffith who has done this. Frederick Griffith experiment of Streptococcus pneumonia. Frederick Griffith, he took a bacterium, that is the Diplococcus pneumonia, which is now known as Streptococcus pneumonia. Streptococcus pneumonia, he took the 2 strain, rough and smooth. Rough one, smooth one secrete a polysaccharide capsule. It protects the bacterium from the immune system of the animals, so it is able to produce the disease. It is virulent type and the mice can die. It produces smooth colonies or the solid medium, so it is called as the S type of colonies. Rough, which is unable to secrete a capsule, it produces the colony with the rough appearance on the solid agar plate, and it is a virulent type of colonies which can't cause a disease in mice, and the mice is alive. So here you can see in the diagram, in the figure, that the capsule of the 2 smooth strains and can significantly their chemical composition. You can see the 2S and the 3S. In 2S you can see a polysaccharide capsule and the cell wall, and in the 3S you can see the arrangement of the capsule. So rare mutation are convert a smooth strain into the rough strain and vice versa. Due to the mutation also, the strain can be converted, the rough strain can be converted into the smooth strain, and the smooth strain can be converted into the rough strain. The mutation do not change the type of the strain. So here we are going to see the Griffith experiment. Griffith conducted experiment using the two strains, that is the 3S and 2R. First what he did, injected mouse with the live S type of bacteria. Now S type of bacteria, so it is the virulent type of bacteria. It can have been caused the disease to the mice and the mice would be dead. The type 3 bacteria recovered from the mouse blood and they were 3S type of bacteria and the mouse was died. Now he injected 2R type of bacteria and where the mice was survived. Why? Because 2R they are the virulent type of bacteria, so they are not capable to cause a disease. So here the mice is alive. No living bacteria is isolated from the mouse blood because the mouse immune power already killed the bacteria. Now he inject the mouse with heat killed 3S type of bacteria. Now when he heat killed that bacteria, means the S type of bacteria are already killed over there. Now as they are virulent type, but now they are dead. So they would be unable to cause a disease because they are already dead bacteria. So mouse here survived, no living bacteria were isolated in the mouse blood. Now at last what he did, he took the live type of 2R, which were the non-virulent type of bacteria or the virulent type of bacteria, and he added with that a heat killed S type of bacteria, S cells. Now he was concluding that the mouse would be alive because already the 3S have been heat killed and the 2R they are been virulent. So here the mouse will alive, but he got a shocking result that the mouse was dead. And what he got? He got the type 3 bacteria recovered from the mouse blood. Means type 3 bacteria was found, which was heat killed. They were found in the mouse blood. We can see here in the diagram that in first the living 3S type bacteria were injected. You can see after several days the mouse were lived and he got the isolate, that is the live 3S. In second, what he did, he injected 2R type of bacteria in the mouse. After several days he saw the mouse also survived, no living bacteria were isolated from the mouse. In third, what he did, he heat killed the 3S type of bacteria which were injected into the mouse. After several days he got mouse was survived because already he have heat killed the bacteria. The bacteria are dead bacteria, so they can't cause a disease. No living bacteria were isolated from the mouse. So here also the mouse survived. But in last what he did, he took the B and C. You can see in the diagram the B and C were mixed and then they were injected into the mouse. But here what happened, the 3S have been able to transfer the genes which the genes which are capable to produce the polysaccharide or which are capable to have a capsule to the 2R type of strain, and the 2R type of strain become the 3S type of strain and they caused a disease in the mouse and the mouse was died. So type 3S bacteria were isolated from the dead mouse. So here he made a conclusion that there is something which was being transferred from the 3S type of strain to the 2R type of strain, and so he called it is the transforming principle. But till the date he was not knowing that from the 4 biomolecules which molecule was transferred from the 3S towards the 2R strain. So here we can see the conclusion of Griffith experiment. Griffith concluded that something from the dead type 3S, that is smooth type, was transforming type 2R to the type 3S. He called this process as called as transformation. The substance that allowed this to happen, it was known as transforming principle. But Griffith did not know what it was and what was the nature of the transforming principle. But he was knowing that there are something from the biomolecules that was transferred. Griffith observed that live S type could kill the mice injected with them. When he heat killed the S variants and mixed them with the R variants, then the injected mixture into mice, they were died. Griffith was able to isolate the bacteria from the dead mice and found them to the S type of variety. Thus, the bacteria have been transformed from the rough type towards the smooth version. So, the ability of this substance to change the genetic characteristics of an organism is called as the transformation. So, this is the transforming principle and the Griffith experiment, which shows that there is something which was being transferred from the heat killed S type of strain towards the rough R type of strain. Next, we are going to see scientists set out the isolate this transforming principle since they convinced that it was a carrier of the genetic material, but what was the carrier, that was unknown till now. So, next we are going to see the next experiment which proved that it was the DNA which was acting as the genetic material. So, Avery, Macleod and McCarty, these three experiments collaboratively worked together to find that it was the DNA. DNA is genetic material. In 1944, after the Frederick Griffith experiment, Avery, Macleod and McCarty published a result of the study that identified the transforming principle of Streptococcus pneumonia. This approach was to break open dead cells chemically, separate the components, example protein, nucleic acid, and determine which was capable of transforming live S pneumonia cells. Now, what they did, the Griffith experiment was continued by the Avery, Macleod and McCarty. What they did, the last fourth experiment, which was the isolates which was taken by the Griffith, they were opened up and chemically checked that which one of the molecule have the capability of transforming, which is the carrier of the genetic information. Only the nucleic acid fraction was capable of transforming the bacteria, so they called it as the genetic carrier. They criticized, noted that the nucleic acid fraction was contaminated with proteins. The researcher treated this fraction with either RNase or the protease and still found transforming activity. But when it was treated with the DNase, no transformation occurred, indicating that the transforming principle was the DNA. So, these are the three scientists, that is the Oswald Avery, Colin Macleod and McCarty. These were the three scientists which worked together to prove that it is the DNA as the genetic material. You can, in the figure number 3, you can see the Watson and Crick also. Watson and Crick, they are the scientists who told us the structure of the DNA. How the DNA, who gave us the structure of the DNA and who explained the structure of the DNA, they were the Watson and Crick. Avery, Macleod and McCarty realized that Griffith observation could be used to identify the genetic material. They carried out the experiment in the 1940s. At that time, it was known that DNA and RNA and protein and carbohydrate, they are the major constituent of the cell. So, they only have to distinguish from this four that which one of them is a carrier of the genetic material. They prepared the cell extract from the type 3S containing each of their macromolecules. Only the extract that contained purified DNA was able to convert the 2R into the 3S. On the screen, you can see the experiment is showing the Avery, Macleod and McCarty's experiment, which in that the 3S cell in liquid culture were grown. They were centrifuged, and 3S, for what this were centrifuged? To take up the cells, to open up the cells. Then they were the heat killed cells and the homogenized cells were recovered. Now, if you can see the first cell, it was treated with the deoxyribonuclease. The second, it was treated with the ribonuclease, and the third, it was treated with the protease. Why these are so? Because they would be degrading that particular molecule. Now the 2R cells, the rough colonies plus DNA is treated with the 3S filtrate, they were shown. In the second you can see the 2R cells plus RNA is treated with the 3S filtrate. In the third, RNA cell plus with the protease treated with the 3S filtrate, and in last 2R cells plus 3S filtrate. In first you can see no transformation occurs. In second you can see transformation occurred, third you can see transformation occurred, and fourth also in transformation is occurring. Now why so? Because RNAs will directly dissolve, will kill the RNA, protease enzyme which will degrade the protein, the carbohydrate it will be degrading the carbohydrates, the RNAs will be degrading RNA, so there will be no RNA, no DNA, no RNA, no protein, no carbohydrate, still there is a transformation. Means these three, they are not the molecules which are going to do the transformation. But in the first, there was no transformation because the DNA's enzyme going to degrade the DNA. If there is no DNA, then there would be a no transformation. So conclusion is that only 2R cells were found. There was no transformation. In second, there were the 2R also and 3S also. In third also 2R also, 3S also, and fourth also 2R also and 3S also cells were present. Conclusion of each were there. Active factor RNA was not there. Active factor protein was not there. Active factor carbohydrates or lipids were not there. Or 3S contains the was not an active factor. It was only the DNA which was the active factor of the experiment. So from this they prove that it is the DNA which is a carrier of the genetic information by using the various enzymes. Due to that enzyme, the molecules which are present in the cell, they were degraded. So that molecule were absent. If that molecule absent, still the transformation was going on. So that was not the molecules which are capable to carry the genetic information. So in next slide, again the hypothesis, the genetic material of the cell, which is protein, nucleic acid, that is DNA and RNA. You can see heat killed S type cells, lipids and sugars, they are also there. Remove lipids and sugar from the solution, proteins, RNA and DNA, it remained. Now we have in protein we have added protease, in RNA we have added RNase, and in DNA we have added DNase, all the enzyme. So proteinase will degrade the protein, so there is no protein. RNase will degrade the RNA, no RNA. DNase will degrade the DNA, no DNA. If there is no protein, still there is transformation. If there is no RNA, still there is transformation. But if there is no DNA, there is not transformation. So it is the DNA which is the genetic material. From this, they concluded that it is the genetic material. DNA is the genetic material which is carrying the information from one progeny to its next progeny. So conclusion is this: the purified highly active transforming principle gave a elemental chemical analysis that agreed closely that which calculated the DNA. Optimically, centrifugally and electrophoretic properties of the purified material were those of DNA. There was no loss of transforming activity upon extraction of protein and lipid. Trypsin and chymotrypsin did not affect the transforming activity. Ribonuclease had no effect on the transforming principle. In contrast, transforming activity was lost following the addition of DNase. So this was all about the experiment of Avery Macleod. So in today's session, what we have learned? We have seen the search of genetic material. We have seen the initial discoveries and the history of DNA, that is the Mendel's experiment and Frederick Griffith, and then the Avery Macleod. We have seen the Griffith transformation experiments and in its conclusion. We have seen the Avery Macleod experiment and its conclusion. We have seen that how Griffith and Avery Macleod proved that there was some transforming principle, and later by the Avery Macleod and McCarty's experiment was proved that they given a chemical evidence that it is the DNA which is transforming from the 3s type of strain to the 2r type of strain. So in our next lecture, we are going to see the Hershey-Chase bacteriophage experiments at conclusion, the Guymias Carmen tobacco mosaic virus experiment, they are the viruses which are having the RNA as a genetic material. They prove that, which we are going to see in next session. Frankel contract and Singer in 1957, their experiment, and later on we are going to see the composition of DNA, the structure of DNA and the RNA. So I think everyone have been knowing this very well. If you have not understand, you can ask the questions. So thank you for the listening my lectures. Thank you.